Welcome to this episode of the Accad and Koka Report. Today we are super lucky to have Ali Mortazavi with us today. Ali Mortazavi is someone that I stumbled upon on Twitter. Twitter has all kinds of not good reasons to be on it. Ali is one of the good reasons to be on it. He's just a fund of knowledge about all kinds of things. He's super smart. You can tell because he is constantly posting chessboards that I can't understand. So Ali, welcome to the show.
Thank you very much. Thank you for the lovely introduction. You just reminded me I've got to stop posting those chess puzzles because they're getting a bit too popular. It's incredible how many people respond to them.
Well, you won't have to worry about me or Michelle responding to them. So Ali is CEO of ETherapeutics. He is, I would say, reasonable to say that he's in the investor space in terms of biosciences, and he has one of the reason I've been wanting to get him on to talk about something to educate us for a long time, but the particular thing that kind of he brought up recently that I didn't realize was that he has a pretty deep insights into the mRNA vaccines that we have, and it's an interesting story, and also specifically mRNA, the delivery mechanism that we're currently using, and he knows a lot about that too. So, Ali, I know, you are a competitive chess player. I mean, you're like are you a grandmaster or a black belt, or what do they call it?
Not really. I actually haven't played chess for maybe since 1994, probably. Right. I'm an international master, but chess is a pretty efficient game. I had to give it up. It was just not a way to earn a living. But it's a weird world. It just seems to follow me and haunts me in everything that I do. For such an old game, it's actually incredibly technologically advanced and ahead of its time. So we can go into some of that, but kind of all becomes somehow relevant.
Yeah. It's interesting that you moved from that space. You're a competitive chess player in your early 20s, and you moved into very quickly, I guess, the funding of biological life sciences, correct?
Yeah, I mean, just a very quick run through. Actually, I started off doing a computer science degree, so I did a huge amount of computer programming, which I literally never used ever again. But actually, like I was saying, I was a professional chess player, maybe from the age of 10 sometimes, but for a few years after that. But as I said, this kind of very old game was actually very ahead of its time. Even in around 1990s, I was using a huge amount of sort of chess databases. So there came a point where everyone's games were stored locally on databases. So your opponents, you could see what they did before you played them. So we were ahead of our time there. And then the next evolution of that, kind of a few years later, was what we call chess engines, where computers could basically play chess. So we would look at databases of our opponents, and we would have the computers help to tell us whether our ideas are good or bad. And then obviously Kasparov had this very famous match with Deep Blue, and he lost. And now even chess has evolved in terms of AI and machine learning, with AlphaZero. So this kind of very old game has figured a lot in my life in terms of computation, data sets. But yeah, I mean, around kind of 1994 or 5 or something, I decided it's time to earn a living, join the financial markets. I did everything in financial markets, in the end, a lot of investing in biotech and tech companies. So that's how I kind of got to somewhere around 2011, where I funded a company called Therapeutics. And then I had a really bad idea to become the CEO of that company. And then that's where the kind of fun and games with RNAs and delivery systems, etc. kind of stuff.
Yeah, I was in the little research I was doing for this. I thought it was particularly telling that you were like, you're a professional competitive chess player, and then you move to this world of finance and hedge funds and whatnot, and that seems pretty daunting. But you were like, this is actually pretty easy compared to chess.
Yeah, actually, I mean, it's funny you should say that. I mean, you think the kind of typical image of people in financial markets, so they're kind of extremely ruthless and hardworking.
Yeah.
But I can assure you, professional chess players much more ruthless and efficient, like a different league.
So I have been you know, and again, this just shows you how behind clinical physicians in practice are. At least, you know, I represent. I'm sure I represent some slice of it. I'm sure there are folks that are much more ahead than I am. But in terms of where we are compared to where the research is, right? The first I heard about is something related to what you did in terms of the silence therapeutics. Was with this medication that some people were talking about, where you inject patients once every 6 months, and by silencing the silencing RNA, you know, the RNA, which is the messenger little piece of protein or whatnot that that allows, you know, a messenger that allows making proteins. If you silence that, you know, you can inhibit something, and my goodness, you can do this for 6 months. So, you know, that was super interesting. It's a medicine called inclisiran, correct?
Patisserin, yeah.
So, but again, and that was the first. I swear to you, that was the first I'd heard about this type of mechanism as a therapeutic. Now, again, so tell me a little bit about how that's evolved, because honestly, until then, all I'd heard about was, oh, you know, CRISPR is going to modify the DNA, and you're gonna get these amazing results based on that. So it seems it seems to this Luddite, like, you know, there's two different mechanisms where you can kind of modify, you know, biology, and this kind of came out of the blue. So can you tell us a little bit about the history of silence of this you know, silencing RNA, and then also what your thoughts are in terms of CRISPR and how the two things compare?
So I guess, where should we start? Yeah, so I kind of got into this in 2011, and significantly less knowledge than even you've given now, and obviously you've got all the clinical aspects. But well, look, to go back, first of all, difference between DNA and RNA therapeutics. RNA is transient. You can redose once every 6 months, 3 months, subcutaneous injection nowadays. DNA is obviously, you know, got the kind of permanency aspect, and we can maybe come to that another time. But in 2011, silence therapeutics, you know, RNA, nobody wanted to hear the word RNA. The kind of the macromolecules, it's just a virus. The body looks at it, there's no way am I letting this thing anywhere near me. People were rightly kind of very skeptical. So, you know, maybe it's actually worth rewinding just a little bit of history, where how this whole evolution of delivery systems, what are called these chemical modifications for stability of any RNAs. So go back late 90s, you know, you've got a company called ISIS Pharmaceuticals, called Ionis Pharmaceuticals. They were the kind of first guys. They came in with what's called a single strand RNA, so literally just an RNA with maybe 18 to 23 nucleotides. And maybe that was the kind of first bit of, let's say, genetic medicine, you know, kind of Human Genome Project, blah, blah, blah. But it was pretty crude stuff. You know, their idea was big injection, think lots of liquid. And, you know, we can get somehow this piece of RNA into a cell. We can, with this single strand, find the target gene and do what you just said, basically stop its function. So the first problem was that what they called naked RNAs, i.e. they're just as, you know, almost as endogenous as you could get, just got cleared by the immune system. It just looks at it and just says no way. So efficacy was just terrible, you know. So you could forgive them. Let's call it version 1.0. Then the next evolution of that is, okay, so, you know, we've got to do something to this construct. We've got to, you know, we can't let the immune system just degrade it as soon as you inject it. So let's find some sort of what we call chemical modifications of each of the base pairs to try and stabilize it. Give it enough time to basically circulate, get to somewhere. You know, we'll come to somewhere in a minute where that place is. That had the downside of toxicity, right? So you got stability, you know, you had all sorts of weird and wonderful chemical modifications, you know, phosphorothioate modifications, fluoro modifications, all the kind of chemistry stuff. So kind of stable, but lots of toxicity. And also, you know, if and when these kind of single RNAs got into a cell, you know, they got into every cell. So, you know, the biodistribution would be, you know, this thing would end up in the kidney and the liver and the gallbladder. So you weren't getting any gene selectivity. So, you know, you wanted to, let's say, stop something in the liver, you'd be stopping that same gene in every other cell type or tissue. So it kind of really was going around in circles. Around, let's say, 2008 or 9, people kind of thought, right, what we need is delivery systems. So we need to basically put these RNAs into a delivery system. So around, maybe actually go back a bit, 2005-6, the world of RNA went from single strand to double strand, which are basically what are called short interfering RNAs, which is what I work with at ETherapeutics and at Silence. Basically very similar to single strand RNAs, but the whole big kind of problem was, look, we need to basically encapsulate this thing, protect it in a delivery system to let it get into a delivery system, we'll put it into a cell, and then kind of, we can basically, have a therapeutic process. And those delivery systems very varied, but lipid nanoparticles, very crude, blunt, immunogenic, inflammatory constructs seem to kind of work, right? So suddenly we were putting at Silence these double-stranded RNAs literally just wrapped inside of a kind of lipid nanoparticle, and it was reasonably successful, and then we ran into problems like 80-minute, 90-minute IV infusions, so a patient had to go to hospital. That's not a great start. Then you had to have steroid pre-medication to try and dampen the immune system down even more. You had antihistamines, everything.
So the lipid nanoparticles, they were just what's their history in terms of using them as a delivery system for?
Just a question of just iteration and trial and error. Lots of different systems were used. None of them were kind of successful. But the lipid nanoparticles, started to show a particular affinity to the liver, which basically anything does. But they were successful in carrying a piece of RNA, at least to the liver, safely without being broken up by the immune system in transit, if you like. Okay, so conceptually that's at least one tick in the box. But then you ran into these kind of
But you're saying that they needed steroid pre-medication stuff. So why? What was it about the lipid nanoparticle that made it so immunogenic? Or we don't know. It is.
They are highly immunogenic.
Okay.
They're charged. They're positively charged. They kind of all sorts of properties that you really don't want. So, I mean, I guess one thing that kind of surprises me is that there's still this notion. Every time I'm talking to Twitter, that I see, whether it's vaccines or anything, well, mainly vaccines, is there's just this now established concept that they're just somehow totally inert, which is just absurd, right? Like they're not, right? Then you get to a point, and we'll come to that in a minute, that dose does matter, but they are absolutely not inert. They're highly inflammatory and highly immunogenic. And frankly, for a long time, the RNA companies like Silence, like us, we did everything to try and get rid of them. It's quite funny to see their evolution as some sort of like life-saving, construct, because we just were so fed up with them. We'll come to that. So, but at least they were getting into it. But we ran into these kind of practical problems.
So the patients would have, just as for the clinical interesting stuff, the patients would have what, fevers, body aches all over, or LFT abnormalities? Like, what would they have?
Well, I think without you'd have to have the steroid pre-medication and the kind of antihistamines. That there would be no options for that. So anaphylactic shock, all sorts of other things that could easily happen. But look, I mean, in 2018, so this is probably why you haven't heard of it. Patisserin, TTR amyloidosis, approved Alnylam Pharmaceuticals. Kind of, it works, but all the problems we talked about. Patients have to go to hospital for every month, 3 weeks infusions.
Right.
It's fine, but it's TTR is a bad enough disease where it can warrant it. But take inclisiran, PCSK9 inhibitors. Forget it. No one's going to go to the hospital.
So just for users, listeners, TTR. We're talking about Patisserin. So Patisserin was the first RNA packaged in. It was packaged in lipid nanoparticle.
Yeah.
Yes, and this
probably
was
not the first, but the first that was
that was commercially widespread success and stuff, and that's something that touched us as cardiologists, because this is TTR amyloid. I mean, for the longest time, we did nothing for patients with amyloid. We would just watch their watch the natural history of amyloid progress, and then suddenly you had trials showing that, especially for as I'm remembering, the neuropathic pain and stuff, there were significant benefits seen to this RNA that was effectively shutting down production of this deformed protein that once it got out in the blood, it would misfold and then deposit in all these different organs, one of them being the heart, which would then cause these issues. So so for the longest time, we had nothing else to do other than give these folks Patisserin. Patisserin couldn't be given in a doctor's office because it wasn't pillformin. And as you're saying, it had to be given in an infusion center. It had to be done on an every 3-month basis. This was the first widely available. And was this kind of like a breakthrough to see that?
No question, a breakthrough. Quite a few hiccups with other clinical trials in oncology with lipid nanoparticles and siRNAs, probably for different reasons. But it was, kind of monogenic-ish disease. It was kind of, liver, etc. And as you say, it was an unmet need. And so, fine, every 3 weeks is kind of not a problem. But you struggle to find other kind of genes in the liver that you could really sort of build around. So, we at Silence, every competitor in the RNA space, we did everything to try and get away from lipid nanoparticles as any type of sort of chronic treatment.
Ali, roughly speaking, sorry, the lipid nanoparticle, is it just an emulsion, a sort of a fancy emulsion of fat, or what's?
No,
how would you describe a lipid nanoparticle?
It's like a sort of, think of it as like a sort of envelope where it just literally protects the whatever cargo you put in it. Actually, you can put an antisense into it. You can almost put anything into it.
Okay.
But it just came at the cost of this, and the kind of positive charge, everything about it was just too immunogenic and too.
But it's a manufacturing process, a way of putting whatever whatever drug you want to put in, or mRNA, with a fatty substance.
Yeah, and then and basically, you could put any kind of cargo inside of it. So it did the job in certain ways, right? And we'll come to why, in terms of vaccines, a lot of pretty amazing things came together in a horrendous setting, let's just say. But in those days of kind of using it for a chronic treatment for inhibiting any target gene, you were really kind of stuck. TTR, I'm trying to think of, oncology became a real problem because nothing really worked. We were, even at Silence, really struggling to find, we'd find good ideas, if you like, good targets, we'd work it up, and then you come to the commercial place and you go, look, this is just like, we haven't got a position here. Like, it's impossible. We're up against all sorts of different sort of competitions, basically. But then, in the siRNA, which is the inhibitor space of the kind of business that I'm in, like most scientific inventions, they don't sort of get highlighted in the newspapers on the day, but something very dramatic happened around 2013 or so, maybe I'm 13, 14, where Alnylam Pharmaceuticals started working with a delivery system called kind of GalNAc conjugate. So this was a very old kind of delivery system which has sort of re-emerged, and it seemed to basically be very specific to the liver. The mechanism of getting into the liver was very understood for a long time. And so what happened, though, was this RNA was now attached to it. So it's gone from being in an envelope out of the envelope, but attached to this very neat delivery system. But being out of the envelope suddenly meant you're back into this world of stabilizing it in terms of chemical modification. So you've got to give it enough time to, from injection, get into the liver and start the kind of silencing process. And we can get into that sort of mechanism a bit later. But the big kind of change with that was, the biological mechanisms are incredibly well understood, receptor mediated, so we knew like kind of how it was being internalized. But really probably the biggest thing was that one subcutaneous injection could inhibit and can inhibit a target gene from 3 to 6, maybe even longer, maybe even a year, months. So suddenly you're not doing every 3 weeks IV pre-medication. Suddenly the whole world of kind of, cardiometabolic diseases, chronic diseases, obesity, everything is open, because once every 6 months is a complete, one very small subcutaneous injection is a, very competitive drug profile, if you like.
So you're talking about the GalNAc, right?
Yes. So that's really specific to the interfering kind of RNAs. So that's kind of, if you like, my world. But I'm just giving you the kind of evolution of whether RNAs, they need to be chemically stabilized in order to basically evade the immune system. Then they're within a lipid nanoparticle delivery system to protect them. Then they're suddenly out of them. And we're back in the world of chemical stability again, because the mRNAs kind of go through the same torturous path and blah, 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 if you like. Okay, so let's just imagine that. So around 2015, 16, Patisserin is on its way to approval, but the new technology is obviously kind of much, much more, let's say, robust in every type of activity, safety, patient adherence. It's just much better. And then around 2014, maybe 13, Moderna kind of pop up, very large funding, I think from Viking or Flagship, with this, let's say, slogan, right? Let's make your cells a drug-making factory. You get a piece of it, get an mRNA into a cell, any cell, and the ribosomes will start making, the protein. Sounds great, if you could do that. And the problem they had was multiples of what the short interfering RNAs had. Number one of just size and length, typical mRNA, thousands of nucleotides potentially, right? So it's like, even what do you do with this thing? It's just very large for a start. And in terms of immunogenicity and, if you like, stability, or, quantum step worse, highly unstable, just degrades just instantly. So, if nature didn't like double-stranded RNAs, it loathes mRNAs more than anything else. Still, they really shifted it for a very, very long time. Actually, in the end, settled for using them within a lipid nanoparticle system, just like we did, right? Because they couldn't attach it to a GalNAc. It was just too long, and it was just, too big, etc. So they couldn't do, an siRNA is whatever, 18 to 23 nucleotides. We're talking about something 1,000. So, fundamentally different sort of proposition. So they're inside of a lipid nanoparticle. They're getting into a liver, which is not bad in animals. So they're turning up. mRNA turns up in the liver. Proteins get produced. But the kind of therapeutic effect is 24 hours, 48 hours, and they just can't stabilize this thing inside of a cell for long enough to basically have it going. So, the entire kind of Moderna thesis from the very beginning was protein production, the cells being a kind of factory. And when they started having difficulties with that, they started to talk about vaccines a bit more, but even they kind of went very quiet. I mean, I would probably say, Moderna were maybe close to an existential crisis. They really had big, big technical challenges, huge. In fact, they haven't solved them. No one has solved them. Anybody who can solve the stability issues of an mRNA inside of a cell is genuinely looking at, a very, very important drug modality, very, very different to, say, gene therapy. Oh, so right. So now we're kind of, pandemic day one, and something sort of terrible events create the most incredible sort of opportunities. So we got this kind of virus, and some, and it's got a spike protein, and they sequence it, and it's like, hang on. So what we can do is we can use a lipid nanoparticle, but we don't have to infuse anyone for 90 minutes for a month. We can just give a very quick shot of it, right? So you don't get any of those sort of potential toxicities. You don't need any of those sort of immune suppressants or this kind of stuff. You can put the mRNA in there, and the mRNA encodes for a spike protein, right? So it's kind of an amazing sort of opportunity that turns up. One injection, the spike. So remember, mRNAs are very unstable. They can't make the spike for very long, but they make it for long enough, like flash photography, that the immune system kind of wakes up and, let's say, takes over and does X, Y, and Z, right? And I guess, actually, funny enough, today when I was just looking up a few things like that, I was thinking the actual mechanism, because look, when you get an injection of mRNA vaccine, it's got to get the mRNA has got to go into a cell in order for the ribosomes to make the spike protein. And I thought I knew how they did that. I was thinking, I don't actually know how they do that. But now I realize, so what it is just the local inflammation recruits kind of neutrophils, APCs, and if it wasn't for that local inflammation, those kind of like immune cells wouldn't be recruited, and they actually just, for unknown mechanisms, the lipid nanoparticle sort of dumps the mRNA in there. Then the spike protein gets produced in those very kind of crude mechanism, and then basically the kind of innate immunity kind of takes over. So I didn't kind of know that it was just, even that is a bit of an odd way of kind of expressing the spike protein.
So it's not the muscle cells in your deltoid that take up the mRNA.
That's what I kind of, I hadn't really thought about it at all. So it's that very crude mechanism, but but it suddenly played into their weak spots. So they didn't need long expression. They needed just flash expression. They didn't need, so let's say Patisserin, I'm trying to remember, lipid nanoparticle dose, something, let's say number like 125, something like that. I can't remember. But and the let's say vaccines around 10 or something like that. So significantly lower dose of lipid nanoparticle. So quick injection, quick bit of kind of flash expression of a spike in a very kind of crude mechanism by local inflammation. If you didn't have that local inflammation, probably nothing would happen. It would just, already I think about 20% of the product ends up in the liver anyway, just gets degraded. So that crude mechanism is enough to get the mRNA in there, ribosome kicks in, spike protein is expressed, and then kind of innate immunity takes over. And then that, I think, is kind of like, a very strange way of reinventing an entire company. But obviously, to be fair to them, I think they even the spike protein is quite even though it obviously mutates and is problematic, but they were able to sequence it so fast and make the mRNA on a computer literally within days, and make GMP material vaccine within 30 days, So everything came together at once to create that, pretty exceptional set of circumstances.
Oh, that's so fascinating. So just to
sorry. What's the story about attempts to use mRNA for vaccines that date back to the 1990s? Right. So Robert Malone now is known to the whole public as the co-inventor of mRNA technology for vaccines. What's that story?
Yeah, I mean, I've seen him say that. No one who I don't know what the inventor means exactly. Right. But I mean, actually, they've had quite a lot of trouble with other vaccines. I think their latest Zika and flu weren't anywhere as good. To be honest, I'm not a total expert, but my guess is it's the spike protein which creates the unusual set of very good circumstances, if you like. Whereas I don't know what they're expressing in terms of the flu and Zika. Probably much more problematic is my guess.
Okay.
So just so there's multiple different RNAs. There's messenger RNA, there's small interfering RNA, correct? And there's many other different types. So those are the things there's so many. Yeah. So that's what we're talking about at the moment. And these folks basically used, the novel thing to do was that Moderna was an mRNA. It was an mRNA-based company that was trying to basically get your cells to produce these things, but they were struggling with how to deliver it and give I guess the struggle is stability and giving it over an extended period of time. And here,
gain of function to be able to make a protein would be, so many diseases would be addressable if you could, but you need more than 24, 96 hours at most of expression. You can't be going backwards. That's worse than sRNA.
Right.
So they've got in terms of protein production, they've got a delivery problem, they've got a stability problem, intracellular stability problem, all sorts of different things. But the vaccines seem to hit every sweet spot. So that's.
Were you surprised at how at the efficacy of the mRNA vaccines, given your background?
No, once I kind of understood the spike protein, I thought. I still, actually now I'm thinking, why haven't I looked up this before? Because this inflammation thing is quite even more if you like, serendipitous or whatever you want to kind of call it. But no, I think now I can definitely see how it can happen. I mean, the spike protein was great because like you could get going fast, but obviously it's not a highly conserved there. It's mutating fast, so you've got a kind of problem.
Right.
But then you've kind of got into this world where people have started to think that mRNAs are almost digital, to be honest. Even sRNAs, it's like, look, you give me a target gene in the liver, I can probably design a drug on a computer in a day, right? But I think you've got to kind of just go a little bit slower. These again, it's highly immunogenic and in of itself, right? So it's probably, that was my biggest worry, is I could see how the efficacy could really work, but I was just wondering about the nanoparticle, about the dose of the nanoparticle, the nanoparticle being cleared, and then I was worried about the immunogenicity of just there's just a lot going on here, right?
Right. And again, the immunogenicity is just kind of a non-specific inflammatory response that has been seen with prior things, but you're giving a much smaller dose in this particular case than before. So whereas you're talking 10, I guess tenfold as much lipid nanoparticle that was going into folks who had who were getting Onpattro or Patisserin, right? And that would provide some surrogate of like how bad, like what were were you aware of what their significant side effect profile was, and was it due to the lipid nanoparticles?
Well, look, we had, like you said, Patisserin is the first. Yeah. And it's a small population in TTR, an indication really. So here we're talking about population-wide vaccines, and it's no surprise that you're beginning to see, let's say, unexplained mechanisms like the kind of myocarditis and other things turn up. It's bound to. The kind of safety and efficacy precedent here was nothing, in real terms. But the dose was comforting. The mRNA component, even that has got quite a lot of, an mRNA is not just, like a template or something. There's kind of many different, even there are chemical modifications there. Actually, to be honest, they've reduced the immunogenicity of the mRNA rather than amplified it, which kind of is counterintuitive. But, like anything, my biggest worry was the population kind of wide things that might creep out. But I wasn't so much worried about the efficacy that I could see work.
Right. What are your thoughts on the adverse event that we have noticed, which is myocarditis in young men? Do you have any thoughts on why that would happen? What the mechanism for that may be?
First culprit, lipid nanoparticle. Whenever things go wrong, I always blame the lipid nanoparticle. But it's almost impossible to say, to be honest. I guess the good news is it's good that almost I'm surprised there's not much more seen in other things that have gone wrong here. So that's the good news. The other thing that I'm kind of very surprised about is, seems to be a reasonably practical solution to it. It's like, just spread the dose and don't give them the donor. I just don't understand why then people kind of make such a big fuss about, even if it's just a solution to it. But it's not nothing. I think when the pandemic first came, you were looking at a very different risk and fear profile. But as things change, of course, like any drug, you start to take it seriously. And then I think, the boosters, they do start to add up, like any drug, you give more things happen, and these things aren't totally cleared. So I'm kind of, say my daughter has just had COVID. She was literally about to get her vaccine, and well, I'm going to save a vaccine dose just because now I can. And maybe I wouldn't have done it, I don't know, a year ago or something. I'm just going with the flow, if you like, right? But I don't know what the mechanism could be. But first culprit is always lipid nanoparticles.
Yeah, that's so interesting, because there's so much. It's so interesting that you can't have these discussions or say things like lipid nanoparticles could be a potential problem, especially given the history of it. And like I said, it was news to me that people have struggled with using lipid nanoparticles in part because they result in a lot of adverse events to patients.
You get into this strange world of Twitter where it's like they start a discussion, and then when you start discussing it, it's like, oh no, you're promoting hesitancy. It's like, well, are we discussing it or not? I always feel like,
yeah,
when it came to it, people were sort of like, oh, there's other people in the room. It's like, well, okay,
right.
Let's get another room, or let's not talk about it, right? Whichever one you want, like people kind of almost want to use it for their own, I don't know, let's say forum. No one else can come in and sort of say this, because they're just always they always have some sort of wild card of, oh no, you've got to be careful not to say that. It's like, well, I don't know.
But on the other end of the spectrum, what about the claims that these things are causing the production of unregulated production of a spike protein everywhere, and now you have sort of a spike protein epidemic, an iatrogenic spike protein epidemic? Does that doesn't sound plausible to you?
I mean, yes, no. The good news is, I would say that kind of, let's say the acute toxicity worries with completely novel, whatever, maybe we got some good, well, no, we do have some good kind of data there. The second aspect is, look, take about the mechanism. This is not, there's a lot of, let's just say, leakage, right? There's things going to the liver, maybe things staying in the liver, so nobody knows. I haven't seen any data, but that's probably virtue of the fact that nobody wants to show me the data. But if I can't guess, maybe also it's one of these things in this wonderful world of COVID where I don't want to know either. We're over measuring like crazy, and I'm not sure what the end game is. I'm sure if you set me a task to go and find something, I would. And if you set me the other task, I'd probably do that too.
Right.
We just kind of like,
Right. But I think the point that you make is that these things are so extremely difficult to understand and study. In terms of really what happens in the body, right? Because there's a plausible mechanism of how these vaccines work, and I think it turned out to be effective, but in terms of exactly what happens, how these, in the body, how these mRNAs are expressed, where exactly they go, once you inject it and so forth, and what are the downstream ramifications and what happens to the immune system long term and all that stuff. That's really impossible to.
Yeah, look, if I was being fair about it, even say double siRNAs with 4 approved drugs, much kind of simpler, in some ways, mechanism localized to the liver. There's lots of things we don't know about drugs, lots of drugs. And the way we resolve that is we have an equation, risk, benefit, and a disease. Right,
exactly.
And so to me, at the very beginning, no one could argue that this is, a new respiratory virus, like, bloody hell, where's it come from? Clearly an age-related, kind of just catastrophic over a certain point. And that has evolved, right? But kind of, you have to go with that evolvement, and you can't ever know everything. But you have to weigh the risk benefit. But people kind of always want to look at the benefits, and it's like, I don't know in which context. The risk benefit for a 75-year-old is different, for a 25-year-old, and also every bit of landscape on natural immunity and et cetera, whether antibodies are good enough, which I don't think, to me, they're just, no one's discussing the equation. They're just discussing almost sometimes just blind efficacy. It's like, well, I know, give more of a good drug if you like to someone, yeah, they'll get more antibodies or something. But at some point, you've got to kind of put it in the context that it's from. Otherwise, what are we doing here? Give me more on anything. I'm sure I'd, do fine.
Where do you think we go from here? It's interesting that in the therapeutic delivery space, you went from lipid nanoparticles, which is thought of as a crude, blunt object, to GalNAc, which is which is basically complexing the RNA, whatever RNA you have, to a specific complex that then binds very specifically some receptor that's sitting on the liver cell, that's highly expressed on the liver cell, correct? So that's a much more robust way of doing that. Is there is there any thoughts that do you think that's how this is also going to evolve? That we're going to move away from this type of delivery system, or are we going to, or is mRNA the problem? Or is there a better way to do this?
When I look at I mean, I've got lots of problems in ETherapeutics, but one problem I don't have is I know that my drug, if you like, goes to hepatocytes, and I'm largely unscathed in terms of cell type. Okay, it does what it says on the tin. It's reproducible. But kind of all these other modalities, mRNA, gene editing, CRISPR, it doesn't matter what it is, right? It's the kind of cargo element, if you like, the cutting, editing, replacing is in inverted commas the easy bit. They're all basically got a delivery problem. In fact, everyone's got a delivery problem, because this kind of GalNAc conjugate, this is just a ridiculous piece of luck, is the honest answer, right? I've not seen, and I've looked at a lot, and there is nothing even close to it, to that type of cell type specificity with all the kind of properties that I kind of talked to you about. So, you take gene editing, a good example, right? So they're playing in the playground of short interfering RNAs in the liver, right? But there's no unmet need to me there, really, and people talk about, let's say, patient compliance or convenience. I mean, I'm sorry, I don't do convenience as drugs yet in my life. I just don't buy it, right? We've got a drug X every quarter or every 6 months. Why would someone go and do something which they have no precedent for in the liver? And the reason they're in the liver is because they can get into it with a lipid nanoparticle as one shot. So they can do it, but the problem is they should really have delivery systems elsewhere. If they had a delivery system to kidney cells, would be game-changing gene editing, So in effect, they're all sort of camouflaged delivery players. There's been some success in CNS, I guess, with intrathecal delivery. It's pretty good, but uptake of ASOs and now RNAi. But again, that's not one cell type. You're going to kind of go to multiple cell types. We don't know where it's going deep enough into the brain tissue. Yeah. So basically, the drug discovery chemistry, we've got a problem with delivery system, cell type specific delivery system.
What's the problem with the viral vectors? That's what Johnson & Johnson and AstraZeneca have, correct? Those are their vaccines, which are basically they use the adenoviruses. They have DNA in them, and obviously adenovirus is very good at getting into cells, and so
to be honest, it's not my kind of area. Again, maybe I've been biased and brought up on two things not to go near anything with the word virus, I don't know, and or the other ones, and lipid nanoparticles. But look, actually, I remember my first month at Silence, so I remember saying, what's good at getting into cells? It's like viruses, right? So it makes an intuitive sense, but there's been a lot of gene therapy blow-ups and oncogenic events, and so again, you see, Everyone's ready and loaded with gene therapies, replacements, editing, fungal CRISPRs, but they're all going to the liver because that's the only thing they can do. And then the liver is, by chance for me or not for them or whatever, extremely well served. You're getting into a place where, I don't want to edit my PCSK9, or do you?
Really. That's a great point, right? So now, again, completely not something that was on my radar, but you have a company like Verve Therapeutics that is promising gene editing for people at high risk. So they will go in and snip and forever change your DNA so that they reduce your lifetime risk of disease with one shot. Why would you want to
doing that in another cell type apart from hepatocytes in an unmet need? Fine. Again, depends on what the disease is and whatever. But gene editing only works because of the delivery in the liver and ex vivo. So you extract the cells, but then that's not the real thing. There's some in vivo data developing.
Well, why would you with small interfering RNAs? Why would you take the chance of editing the genome? Like, why would you play around with? It's like me.
Well, there's two aspects to it. First of all, I wouldn't, because I would prefer the kind of transient option, even transient and long transient, reversible. And the second aspect of it is, sometimes I think our knowledge of biology is so crude. We're obsessed with maximal inhibition. And there's nothing to say that I want to just destroy TTR, for example, even, for example, I think retaining X% expression is the right thing to do. Right? No one knows how much, right? But, 19, it's not an oncogene, not that I even necessarily believe in that, but they're not oncogenes. You don't want to just flatten them necessarily. Even a monogenic has got multiple functions. So, you're kind of taking, I don't see a commercial proposition, but the data on face value looks great. That's what I will say that, right? And it makes beautiful use of the one-shot lipid nanoparticle again, so you don't have to go and do this monthly that we used to have to do in the SRA. It's once and done, if you like. It's once and done, but not for me.
That's so interesting. So is your skepticism of that space, skepticism of, single gene editing, is that reflected in funding? Meaning, is there a vastly more funding in the biotech space that's going towards, things like small interfering RNA, things that are, and not as much going towards,
There was, but no, the world of biotech funding has moved very dramatically. I don't think the same would happen now. Novo Nordisk took over an sRNA company, Dicerna, in November, let's put it that way. So, for $3.3 billion. But look, companies like Beam and Verve that, doing extremely well. So Beam's trying to resolve some of these. They've bought a kind of very fast throughput lipid nanoparticle technologies testing system. So they're trying to find other cell types. Okay. But for me, I just don't think they'll find the cell type specificity. Because remember, edit a gene, you can only be in that cell type. You can't, you'll probably be okay with PCSK9, I don't know. But I don't really want to be sort of contaminating, I don't know, the kidney, et cetera, et cetera. I've got to be in one place, right? So editing and specificity comes with a limitation of specificity.
So interesting. So right now, the space that we're talking about is liver focused, because that's all we can really get to with some level of specificity.
Basically, the lipid nanoparticles and GalNAc are the only things that get to the liver.
You've got so until we have
CNS, but it's
Right.
But doesn't look good enough to me. Then you do have the eye, I guess, but even that is difficult.
Right. Yeah. No. So interesting. What? So we'll move on a little bit. You are now CEO of ETherapeutics.
Yep.
And you'll have to explain a little bit about it, but or you'll have to explain what exactly prompted the move and what the value proposition you see there.
Where should we start? So Silence, 2011, a long time, lipid nanoparticles, GalNAc conjugates, chemistry, a lot of, this whole RNA space is very kind of chemistry led. All the participants, if you like, and people are very, very, chemists more than they are biologists, because, they were trying to get something to work. I left in 2018 and remember thinking, we've got something to work and it's really good, but kind of, no one knows anything about human biology. Biology is a great unmet need, and the kind of easy monogenic diseases had been taken by competitors. So we had to start thinking about biology differently. And I bumped into ETherapeutics. You're in the kind of world of computational biology, but also liked the fact that they were looking at biology from a systems perspective rather than, really, I wanted a strategy towards biology because I feel as though there's some very simplistic kind of thought processes about biology, if you like. Right? So ETherapeutics, what I was surprised about is how the field of computation had, since my day of doing computer programming, taken off so much in terms of machine learning and kind of, big data and stuff. And these are kind of big words, but there were, some big things happen. Like, for example, Amazon Compute, dynamic compute, we can rent unlimited, for our purposes, compute power. 7 years ago, we would be doing things on a PC, or 7 PCs. So the dynamics had materially kind of changed, if you like. And then I thought, if I could bring this kind of GalNAc technology and cell type specificity to the liver, we could kind of zoom into the liver, if you like, computationally. So it became, if you like, the liver hepatocyte biology coupled with a drug technology which works, basically. And then, things became maybe a bit more sort of rational and chess-like, if you like. And that's the way I kind of was in my mind looking at it. So, ETherapeutics had a sort of still has a systemic computational platform, but we've kind of at least zoomed into one cell type. Okay, it's not quite sort of localized. There's a lot going on kind of in terms of circulatory proteins, but at least we're in kind of one tangible place. So we're now creating the biggest data resource, biological resource of hepatocyte biology, right? To try and look at biology kind of differently, more sort of systems, networks, because I think that is really where there's an advantage, big advantage to have. So, liver, the siRNAs in the liver aren't just for sort of liver diseases or liver fibrosis or NASH. One of the biggest markets is obviously cardiovascular diseases because of the kind of secreted proteins, PCSK9s, ApoC3s, etc. So you're not, by going to one cell type, your market size, if you like, is by no means limited. It's actually very, very big. And for me, I go back to the one thing is the drug technology works. It's got proven efficacy and safety, and a lot of people, anything that works in this industry, take it. Don't change it. Whereas other competitors are trying to find other cell types and to try and open up new frontiers, which I kind of understand. But so we're zooming into biology, computationally zooming in, trying to model biology, if you like, trying to sort of find ways that we can try and fail early, fail kind of cheap, if you like, and do things in a as much as possible, fast throughput way. And other people have, I think, kind of migrated the liver. It is competitive, but I think there's more to it than monogenic diseases and PCSK9s.
So you think that there's how do you validate this stuff? Meaning, it sounds like you're trying to go from big data to coming up with something that works, right? That's, I don't know, that's the million-foot view. how do you generate? How do you validate that? How do you know? I mean, how do you know that what you're doing in terms of? So I guess you're using some type of AI, machine learning, etc., to crunch a large number of data to then come up with novel drug/delivery systems, correct?
Yeah, I mean, it's sometimes, actually maybe even large or big data is maybe the wrong way. Small data sets, which are better, are just better, right? You don't always find things in big sort of things. But yeah, I mean, I guess the starting point is we're in one cell type. We know that if it comes to it and we find a target, our drug technology works. And the second aspect of it is, ETherapeutics was very focused on small molecule drug discovery, which is completely fine. But from a target to, let's say, biological readout in vivo, at least, siRNAs are extremely fast. We're talking, 15, 18 months if you kind of really project plan it properly, which is lightning speed in this industry compared to small molecules. And, so you can basically, drug technology which works, you can go very, very fast, and you can kind of get at least a good biological readout, even if it is preclinically, in record time and kind of very small amounts of money. And the next aspect of it is, I mean, look, this is a completely emergent industry. It's fine having loads of data, the best machine learning algorithms, but how do you go from A to B to C? And that's our major project. We try and model biological processes computationally, so kind of protein to protein interaction networks and systems where we think that, we've done it enough times that, if it's produced X computationally, then, it gives a degree of confidence kind of going forward. I mean, I'll give you one practical example. We did a collaboration with Novo Nordisk and type 2 diabetes, really robust piece of work. It went to the back, kind of in the catalog, if you like, right? And about a few months ago, our kind of team was, doing this research as it does or whatever, and we came across a one or two papers with a new potentially genetically validated target for type 2 diabetes. It was kind of very interesting. Referencing it back to our work and the assay work we've done, was like, oh my God, we didn't even spot this. It's kind of very high on our list, right? You know, those are the sort of things that kind of putting together information, then, you know, having the best early assays, because again, you know, we're in hepatocytes, so at least we can test a lot of our algorithmic hypotheses in primary human hepatocytes. It's not, we're just trying to narrow that gap, you know, trying to get that bit of de-risking, whatever it is, 10, 20%, 30% reduction, you know, eventually there'll be organs on a chip. Not when I'm here, where you can really hypothesize, like, you know, like I used to and now do, you can do when I play chess. I put a chess position up, I press engine, within 5 seconds, it's told me everything about it. Because the chessboard is a perfect model, if you like, 8 by 8 square, 32 pieces, and all the rules are defined. Now, that's a very simplistic way, but we're looking for pockets of those where we can model those computationally. Oh. And the drug technology kind of is helpful, because before you could do all this work, but if it takes you 4 or 5 years to make your small molecule, it's undoable for a small company.
Right? Fascinating.
Yeah.
Ali, do you how do you, like, what's the timescales that you have for, you know, success in terms of what you have to churn out that's something that's of value that's to somebody else in doing work like this?
I mean, I think, look, there are kind of, again, the RNA space is, even despite this kind of catastrophic stock markets at the moment, is particularly wanted because it really is a new validated drug platform, you know, they don't come along very often, monoclonal antibodies, RNA in the liver. So we've got that kind of advantage. And I think we, you know, kind of at the starting point, we even now really have the biggest resource of hepatocyte biology computationally that exists. So you want to know anything about what goes on within a hepatocyte, the disease within a hepatocyte, regulatory genes, DNA, RNA, omics, genomics, you name it, patents, they're all basically within our kind of grasp. So we think even that data set in terms of partnering with people. So, you know, whereas maybe when I was at Silence, they used to say, how good is your technology compared to competitors? We'd say, oh, as good. And they'd say, well, have you got any good ideas? We'd be like, you know, not bad. But now we come to the table with 2 components of the kind of core biology, computational biology data sets and algorithms and assays, and we come with the drug technology. So there's partnership opportunities there. And the second part of it is that in the RNA space, at least, if you show biological activity preclinically, you do get paid for that. There's many precedents for doing that. You know, maybe because of that kind of translatability of genetics, if you like, you know, you've got the same sequence potentially in mice, primates, and humans. So they will pay for that, and obviously the kind of hockey stick returns and phase 1s are also kind of open to us. So we can play that sort of hybrid role. But again, so from target ideas, so if you came up with a hepatocyte target for me now, we would be up and running with the final drug within, let's say, 6 months. You know, and we're ready to go into models and whatever, and we, you know, within 15, 18 months, depending on disease, etc., we have got the drug that could be the same drug if it gets approved, and you've got biological readout in 2 models, etc. That's a big advantage over kind of very elaborate other systems.
Fantastic. What do you make of the recent? We'll end with what do you make of the recent bloodbath, if you will? That's going. You know, so there was is this a correction, or is this something that reflective of something?
I mean, it's difficult to say, because, you know, you've got kind of Ukraine on top, you've got inflation on top, but it's kind of odd. Maybe it's a lagging indicator, but what I can see in the labor market, I can't hire a chemist, oligochemist to save my life. That's impossible. But, you know, people are pretty hard to hire, and they're still, you know, the labor market is very tight, the private market, you know, so it doesn't feel like there's just some people are giving up on global kind of healthcare.
Yeah.
I think, but it is going on for a while too long. There's some very kind of everything has come at once, and there's some very big structural problems in the US biotech market. A lot of hedge funds have, a lot of companies have got bad shareholders who can't hold them, and they just have to sell them.
Yeah.
There's been a huge amount of deleveraging. Lots of technical factors, I think.
I wonder when folks will start to get worried about the ability of the United States to continue to, the U.S. is the biggest market for all these drugs, right? Every drug every little lab in Turkey or India that's doing some type of basic science research is kind of looking for a payoff that comes from the American taxpayer in some ways, right? Yeah. Because of how massive that is, and of course the U.S. debt continues to balloon to some crazy numbers. So I wonder when folks will start to get worried about the United States ability to
I think pay for things. That's a big problem. So then actually we've got some good ideas towards that in terms of pricing and drugs. But, I think one of the main problems with the drug industry is sometimes I think people kind of forget that drugs actually have to perform just like cars and restaurants. Like I know they're drugs, but how many great drugs have there been kind of recently? Like you're kind of getting pretty marginal,
seeing lots of 10,000 patient trials to show some little benefit at 5 years that but anyway those folks
maybe the regulatory process, the whole everyone's going to kind of work together, but I do think the drug industry has suffered from a kind of, I think human biology and knowledge of it is the biggest problem, because everybody goes for the same validated target. So they kind of, it just kind of feeds nonsense, basically.
Given the conversation on Twitter, though, who really needs drugs? Who needs new vaccines? We could just all wear N95 masks and pandemic over, right?
I guess so. if I can't see it yet, but I can't. I've given up arguing because everyone just keeps shifting the goalpost.
Right. I'm flummoxed by some of the. Anyway, but Ali, you've been so kind with your time. Thank you so much.
Thank you for having me.
Absolutely, learned a ton. Your Twitter handle is AA Mortazavi, Mortazavi. We'll have it on the show notes as well. And you are definitely one of the must follows. Super interesting to see what you tweet, and also your conversations back and forth with folks as well is really interesting. So
thank you for having me. And, have a good day and evening.
Thank you.
Bye-bye.